achievements of the Sri Lankan chemist in the context of scientific research at the international level. Today, we are delighted to have a very special personality with us, that is Professor Gomika Udugamasooriya. I would like to warmly welcome you, Professor Udugamasooriya. We are very happy to have you with us today. Without further ado, to introduce the speaker, I would like to invite Dr. Thelma Besinha. Over to you, Dr. Thelma.
Thank you, Dr. Didosha. So it's my great pleasure to introduce Professor Gomika Udugamasooriya for today's webinar. Professor Udugamasooriya is from Kalutara. So while he's actually now in USA, he received his primary and secondary education from D.S. Senanayake College, Colombo, and he's a father of 2 daughters and a son. Professor Gomika Udugamasooriya obtained his BSc degree in 1999 from the University of Colombo, and later on he followed a PhD in the Wayne State University, Michigan, USA. Then he completed a postdoctoral fellowship in University of Texas Southwestern Medical Center, and then thereafter he moved to different positions, and now he is in the University of Houston, MD Anderson Cancer Center, Houston, USA, where he serves as a tenured associate professor of medicinal chemistry. Professor Udugamasooriya is a vibrant scientist and directing a multi-billion cancer research programs mainly focused on cancer drug lead discovery and diagnostic tool developments funded by various funding agents, including NIH and Department of Defense, Cancer Prevention and Research Institute of Texas, and many more. He has published more than 36 research journals so far in widely accepted international journals, and also he has presented over 75 abstracts and introduced a novel 2-color high-throughput drug discovery technology and has 8 international patents so far. In addition, he is an editorial board member and a reviewer of many international scientific journals and member of many grant panels in the United States Government National Institute of Health. He has received many awards, including Chancellor's 50 in 5 Scholar, Faculty Excellence in Research, as well as NIH SPOR Cancer Excellence Career Development Awards, and in his undergraduate career, graduating as a batch top with the gold medal in chemistry. Professor Udugamasooriya has been working in the fields of medicinal chemistry and chemical biology for more than 2 decades, where he is going to share his experience today with us. And he has a research group which is successfully identifying and validating many peptoid components, so far mainly focusing cancer, COVID-19, and other immune diseases. Professor Udugamasooriya is a chair of the Graduate Education Committee right now, and also he had been an advocate for many young scientists coming from Sri Lanka as well as both in United States. And he's an artist and he's a philosopher as well. He is actively engaged in conducting seminars and workshops worldwide on cancer prevention and research, highlighting the value of practicing holistic medical approaches. So without further ado, it's my pleasure to invite Professor Gomika Udugamasooriya for today's webinar. It's over to you, sir.
Thank you very much. Thank you very much for that kind introduction and also for this invitation. I'm very, very happy to interact with my fellow scientists in Sri Lanka. So give me a second, I will share my slides, and I hope you all can see my slides.
Yes, we can see it.
Okay. All right, so so I have been told that this webinar series is mainly to inspire and motivate young Sri Lankan scientists, and and asked me to share my story. I'm very happy to do that. So today I'm going to talk to you about mainly actually my story, and then within that I will talk about the main new technology that I have developed, which has been used now in many places in US and also worldwide to expedite the drug lead discovery process. And I want to tell you that since the time is limited, and I'm trying to show you some of the examples, and later on when I talk about the data, there are many slides and I will run through them very quickly, but I will just mention what is in that, but I don't have much time to go through all the details. All right, so when I look at this poster that was created to introduce my talk, if I look at the very bottom, that's a very nice saying. This was from Marie Curie. Nothing in life is to be feared. It is only to be understood. Of course, definitely. And this is one of my mottos as well. We don't need to be feared on anything. I mean, that's actually that's how I have been doing throughout my life. I really want to explore things, move forward, come forward, and go for it. That's how I was throughout my life. And especially as a researcher, you all need to remember, you always need to question. You always need to question the world and try to understand it. So nothing to be feared. So let me start my journey, talk about my journey from time I had at the University of Colombo. So we all know after the second year, we are being given a chance to, if we are selected to do a special. So I have been selected to all 3 subjects in the biology stream. So of course, I love chemistry, and even in the school, I have been doing projects. So I chose to do chemistry special. And I'm telling you stories even inside my story. So there are take-home points for that. And so I can still remember in the first year, lecture is on organic chemistry mechanisms, and that was done by Professor Dilip De Silva, one of the greatest organic chemists produced by Sri Lanka, world-renowned professor. And of course, because of all this, as students, everyone is afraid of him, and we heard like he's calling students to the board. So everyone is sitting back, and then I wouldn't say I'm fearless, but I was sitting in the front, and then he started the lecture, and within 5 to 10 minutes, he was getting into this esterification mechanism, and he asked the question, and then no one is answering. So I started answering, and immediately he asked me to come forward and gave me the chalk and asked me to write the mechanism. And first, actually, so I draw this lone pair and look at him. He was happy. And then I drew the first arrow. It looks fine. So he was happy. And from that moment to this exact moment I'm speaking, there was a great relationship built between 2 of us. And I did my research project with him. And beyond that, he had helped me a lot. He actually shaped my career and actually direct my career because he encourages me to come to US, and he even personally helped me a lot doing TOEFL and all other things. So the take-home point is, if I was also sitting on the back end without coming forward, of course, I may end up getting 4.0 GPA, but I wouldn't have learned a lot. And I wouldn't have got this kind of really great help from this type of great professors. So don't be feared, and come forward and be active and move forward. All right, so then I came to US for my PhD and at the Wayne State University. And this is how our my first meeting with my thesis mentor Professor Mark Sparler. And this was the question I had to him, my first question: can we develop a drug? And you can see my face, very excited, enthusiastic, and really into him. And look at his face. He has kind of looked at me like seeing an alien falling from the sky, right? And he very kindly told me, hey, we cannot develop a drug in academic institute. So of course, and we learn what is the modern drug discovery process. And this is it. First, we identify compounds targeting disease-making biomarkers at the biochemical level in laboratories, and then test in cellular levels, and then go to the animal levels. And then if everything is fine, it goes to the clinical trials, 3 clinical trials, and then finally become a drug. So after putting 10,000 active compounds into the system process, only one become a drug at the end. And you have to spend $1 to $2 billion, about 10 to 15 years. So this is the very complex, the long, detailed current drug discovery process. And before I move forward, I want to take a minute to tell something not related to this talk, but last few years, there was a Facebook post. There were multiple posts circulating saying, I found a cancer drug, which is wrong and also misleading. So of course, I have multiple patented compounds going through this process, and I'll tell you all of them and show them where are they. There are a lot of advancements, that's good, but I have not developed a drug. I never said that. And please don't follow those posts. And I just wanted to mention that. Okay, so this is, and then in my PhD work, we have been working on this, targeting these protein-protein interactions and PDC protein domains. This is the crystal structure we solved with one of my drug lead compounds that I developed. So remember, we call these drug leads, and then they become drug candidates, and then finally become a drug. So, and I'm not going to spend time on this. And then after that, I was so lucky to get a very high-level postdoctoral positions in many places. And finally, I accepted one from University of Texas, Southwestern Medical Center at Dallas, Texas, which is one of the topmost medical centers, harboring highest number of the Nobel Prize winners who are still active. There are about 2,000 faculty also there. Again, now this is my first discussion with my postdoctoral mentor, Professor Thomas Kodanek. So he was actually the director of the Translational Research Center, had about 30, 40 people in his lab, a very famous big scientist. And again, you can see, this is my question. By this time, I know very well, asking can we develop a drug is a foolish question, but you can see, I never gave up. I really wanted to do something in this area, because if you look at those statistics, right, that's a very long time, a lot of problems there in the drug discovery process. So I was asking him, can we accelerate the drug discovery process? So he looked at me and he directly asked me a question: whom do you want to be? What do you want to do in the future? And I directly said, I want to be a person like you, so in the academia. And then he was happy, and then he spent about 2 hours of his time and showed me his whole research program, and then asked me to go back, spend about 2-3 weeks, and then develop something new, brand new, come up with a great idea and see me with the plan. So you all know, we are very excited, motivated. I went back. Within about 2-3 days time, I put together some project plans and went back to see him. And he immediately asked me, I asked you to come back in about 2-3 weeks. Why you came this soon? And I thought, okay, I can show my colors, right? And I have plans, and I showed him. He looked at it and he again asked me, what do you want to do in the future? And I said, yeah, I want to be like you. And then he told me, he threw those ideas and he said, oh yeah, this will do something for you. You will get papers, but if you want to be sitting in a chair like me, you have to come up with something completely brand new, a new idea. Then actually I started realizing what he was telling me. So again, take home point, don't try to, I mean, you can of course do things and move forward, but always try to do the best, highest, the most effective things, especially in research. Okay. Now, again, so I went back and I said I always have a problem with this current drug discovery process, and this is another facts I'm showing. Over the time, this blue line shows how much money we spend in research and development. It's increasing and increasing and increasing, and these dots showing how much drugs coming out. You can see it is not proportional. It is not following the amount we spend. That is a problem for me. And then this is again the same thing in a different way. This shows the new drugs per $1 billion spent. Now in 1950s, there were many drugs coming out, but it go down, down, down. Now it is only about one drug coming out per about $1 billion spent. Now this is another big problem. Why is this? Okay. In drug discovery, the conventional approach is, and now I'm showing a cell surface in a disease cell. These are biomarkers. So what happened is the biologists identify the disease-specific biomarkers, and then the chemists and then the other even biologists come and develop drugs targeting these biomarkers. And then only you look the whether it will affect the normal cells or the side effects. So these off-target effects are the biggest things in this case. There are many other things, the PK/PD properties, biodistribution, and all the other things, metabolic reactions. But off-target effects is one of the main things. So we spend a lot of time here, those 10,000 compounds, and then over the time we find when we go to the complex systems, there are off-target effects. Okay, then I thought, how about bring both these together? If we factor in this off-target, avoiding off-target effects, or the normal situation into the initial drug discovery process. And I was thinking about that. So this is my the biggest approach, and then my whole career is built on based on this idea, and this is what lead to develop this two-color technique, and that I will explain in a minute. Okay, so we are targeting cell surface biomarkers, and they are important in various diseases. Those can be protein, lipid, and carbohydrate. Now, protein targeting, protein biomarkers, hormones, receptors, signaling molecules, transcription factors, and those are the conventional drug targets. But how about protein, the lipids and carbohydrates? There are not much techniques to target that. Okay. There are two biochemical tools I want to introduce. We are working on this molecular class called peptoids, and then I will talk about this technique. So there are two major drug discovery process. The structure-based drug design, of course, we all know. The rational design, it's like cutting a key to the keyhole, and then working one molecule at a time. It's a long process. The second approach is the combinatorial high throughput drug discovery. In other words, bring all the keys and let the keyhole to pick the best one. So develop large chemical libraries and then screen against the biomarker and then get the best one. We can also attach diagnostic agents and use it for diagnostic purposes as well. So these are the three major classes of compounds, actually drugs in the clinic, small organic, of course, the most famous small organic molecules, and then peptide drugs and also antibodies. The compound class that we are working on is actually the peptoids, derived from peptides to avoid the weaker qualities in peptides. So the peptides, you all know, build based on the amino acid chains. The N-terminal, C-terminal, and the alpha carbon has this R group. There are 20 amino acids. Now in peptoids, this R group has moved to the amide nitrogen. Because of that, these peptoids are serum stable, which peptides not, and then non-immunogenic, it doesn't produce immune signal, and more cell permeable, highly diverse, synthesis is rapid and economical, very easy to optimize, and reported as great protein binding ligands. Now, this is actually Professor Ron Zuckerman, that he is the inventor and called father of these peptoids. He invented this and introduced about multiple decades back in 90s. And then the Professor Thomas Kodadek, my postdoc advisor, he considered as the second father of these peptoids, because in early 2000s to even now, he has showing this the biological applications of this peptoid. So I am extremely lucky to interact with both these fathers of this molecular class, which is a very field now in the worldwide in drug and other ligand discovery. Okay, now in the synthesis, we don't do solution synthesis, we do solid phase synthesis. These are resin beads, active centers ready to react. Now in peptide synthesis, what we do is we attach one amino acid at a time and build the sequence and then cleave it off to get the compound. Now in peptoid synthesis, as I said, there are active centers ready to react. There are a lot of them in every bead. These are about 150 micrometer diameter. You can see in a naked eye. And so this bromoacidic acid brings the 2 carbon, and then this bromine is substituted by this amine, and then introduce one residue onto the bead equivalent to an amino acid. And then we go through these two again for the second residue. It goes like that. Now there are two unique things. Both these reactions can be done in microwave assisted synthesis, which is less than 1 minute. And then this R group comes from an amine. So there are thousands of amines. So that means we can bring a lot of diversity built into these molecules that peptides don't have. There are only 20 amino acids. So the chemical diversity is really high. And I mentioned at the beginning, we are working on combinatorial high throughput synthesis. So that means we develop large number of compounds. How do we do that? So we use this very interesting split pool synthesis method. If I briefly explain it this way, if there are 3 amines called ABC, we can take these beads into 3 reaction vessels. There are a lot of beads in each. We can couple A here, B here, and C here as the first residue. And then we mix it thoroughly and then separate them back to the 3 vessels again. Now we have all these 3 mixtures in every vessel. And then we couple again A here, B here, and C here as the second residue. Now immediately you can see we already have 9 combinations, 9 different compounds in these beads. So if we have more monomeric units, more steps, we can go very high. So this is one of the libraries we developed. 8 different amines, 6 steps, it's 250,000 of compounds. So, and this can be done within a few days time. And this is another library, about 400,000 diversity. And there are many libraries. Okay. All right. So now when we have these libraries, which end up with 1 bead, 1 compound library, which means every bead has a unique sequence with a lot of copies. Different beads have different compounds, right? How do we find the best drug lead? So the conventional method that has been used about 2 decades when I started working on this was your drug targets are mainly proteins. So you take a genetically engineered protein and purify it and with a tag, and then simply mix it with the library. And then I hope you can see when it binds to a certain peptide or peptoid, it will light up. So we take out that bead and remove other things and then do mass spec sequencing to identify the sequence. So that means we have a drug lead to target this protein, which should be important for a disease. This is how it has been happening. Now you can see, we find this, but the limitation is we don't know the off-target effects. And of course, that's what most of the drug discovery process is going on. That's how you look at the off-target effects later. Remember, that's what I was trying to address. And also this cannot be used to target cell surface receptors easily because you cannot overexpress them. So my idea was to have these receptors on the cell and then expose the whole cell to the library, but not blindly, with a control. With the control cell line, the same cell line, but without that targeting receptor. Now, for example, when I developed this at the beginning, I used this VEGF vascular endothelial receptor to a protein receptor, VEGFR2, which is important in cancer. Now, very briefly, the tumors cannot go beyond few millimeters without the blood supply. So it secretes this VEGF hormone, which comes and binds to these VEGF receptors near the that are on the nearby blood vessels, and this signaling asks these blood vessels to form new blood vessels towards the tumor, bringing nourishment. This is called angiogenesis, and then the tumors can grow very rapidly. Now there are drugs, Avastin, and there are many other to block this interaction, and I use this system to develop this assay. So I want to target VEGF receptors. What I did was I took a cell line, divided into two groups, overexpress this VEGF receptor in one group. So that means, let's say there are 100 common biomolecules in this cell surface, and this has that 100, and this has 101, which is the additional one is the VEGF receptor that we want to target. And then I did a very simple kindergarten thing, which is stain this receptor targeted cells in red. This is in green, mix 1-to-1 and expose to the library. Now I'm looking for only red cell bound bead in the presence of green cells. What does it mean? It means the compound on this bead only binding to this VEGF receptor. And if it that's what we want. And if it binds to any other cell surface, remember, if there are 100, if they bind them as an off target, they are found in these green cells too, and then we will see both red and green bound. Now these are the actual fluorescence microscopic images, and this is what we are looking at, the red cell bound bead, and these are the off target binding compounds, where the green also there. Now, until I developed, as I mentioned, about 2 decades, the drug discovery was going, picking up all these beads because this green control was not there. And then these are off-targeted compounds. We get about thousands of them. And in normal high throughput screens, people who work on this know this, you get like thousands of hits. And then after validating over another many years, after spending a lot of time, you find these have off-target effects and they are not good. And you throw away 99.999% of these drug leads. But with this screen, with this control, we can pick not only the binding, but the most selective, more specific ligand in the very first day of the screen. Again, and the take-home point about this is, now, I did only play with these two simple 2 colors. It's a very kindergarten thing. And I always tell the young scientists, okay, you have a BSc or you may have a PhD and you are a big person, but think simple. If you want to touch your nose, you have to do this directly, right? If you have a PhD, so therefore I want to do big things, don't try to touch the nose this way. Don't try to big things. You can try big things, but think simple. This is the thing, and very simple idea. Okay, so actually in this screen we found about 5 beads, 5 hits out of 400,000 compounds we screen. Actually in my lab now we can do 100,000 compound screens per day. This is very rapid. And at the end of the day, we have a really selective hit for our most important target. So this is one of the compounds, and I'm actually summarizing this whole project in the therapeutic applications. We have published a lot of papers, and we found this compound actually blocks the tumor growth in mice up to that level. And then in the imaging applications, we attach contrast agents and then use for diagnosis in both PET and MRI. So this actually kind of summarize how we find compounds and what are the applications. Okay, all right. So after that, so I have been very successful, and it was completely my idea, and all this development came. And then I was within 2 years of time, I was ready to go for my independent faculty position. So I actually got multiple offers from multiple different institutes in US. But then at the UT Southwestern also offered me a faculty position. So I stayed there. I started my first research group at the UT Southwestern. But then a few years time, I was able to get another position, which is in Houston, in the University of Houston, and also joint appointment with MD Anderson Cancer Center, which is the largest cancer center in the world. And also altogether, this is called with few other institutes, Texas Medical Center, which is the largest medical center in the world. So now I'm there. So this is some of the pictures of my labs in my independent career. All the chemistry facilities, the automated synthesizers, and then I also have my own mass spec, other chemistry facilities, and the cell culture, fluorescence microscopic, plate reading, and all these instruments are in several. I have several labs. So okay, let's get into the science again. Now, so far, what I have told you is the conventional drug discoveries. Okay, biologists first identify the biomolecules, the biological targets, and then chemists come and develop drugs, and then look at the normal off targets later. And then in my first development, I built in this control also into the drug discovery. Now this is again going behind known target. Now the genome, the human genome is resolved, so we know basically all the targets, but there are a lot of other things still unknown. If we wait until biologists finding targets and then go behind that, we are biased. So in my independent career, what I was trying to do is not only identifying compounds for known targets, but try to identify new targets. If there are new biological targets that are important for various diseases, especially for cancer. So the idea is compare the disease cell, take one step back from molecular level to cellular level, and identify compounds targeting the disease cell, in this case cancer cell, compared to the normal cell without knowing the target. So we try to find something found in the cancer cell, and that can be a protein, lipid, carbohydrate, or anything. So that's the cool thing here. We target not only proteins, the lipids and carbohydrates also can be. And there are no direct tools, very good tools to target these. But over the time now, we know there are a lot of lipid and carbohydrates also important in disease progress beyond proteins. Okay. Using this technology, actually summarize everything that we have done. We have already identified actually 7 different brand new drug leads targeting various proteins and 1 lipid. So these 8, all these are patented compounds going through this drug discovery process. And also we are working on in the imaging applications as well, diagnostic side, and unfortunately I don't have time to get into that area. And also combine these two to develop molecules that has therapeutic effect, but at the same time, we can follow the activity, which is real-time therapy monitoring, and very, very important in cancer. And basically the idea is to build these diagnostic agents into the initial library, so we can identify molecules that has both these components. And actually, I have multiple different grants, and this is one of the grants I got from NIH for high risk, high impact for this idea, which came to the top, actually the first percentile. So once again, if you come up with proper ideas and proper plans, you will be benefited, and you can actually make it happen. So basically, in my lab, we are mainly targeting cancer. So the tumors are very complex. It's not like just a cell mass. So one of the main things we are looking at is the biomarker heterogeneity. So if we take this receptor, we can come and develop a drug. Even the best chemist in the world come and develop a drug to this receptor. If this receptor is not found in every cancer cell, that drug is useless. So that's one of the big problems in the targeted discovery, the complexity of this biomarker. We are trying to address that, and then the signaling cascade complexities. Now, if this is a growth signal, and if we try to block it here, it will simply bypass here. So that's another thing. And then the cancer stem cells. They are the most stubborn, notorious cells, small subgroup in the tumor. If you try to get rid of all the other cells, these will remain and bring back the tumor. And there are no proper biomarkers also identified. And then we don't have proper ways to do therapy monitoring. So these are the big problems we are trying to address in my research group. And unfortunately, I don't have time to talk all of this, and I'll talk only about few projects as examples in this second part of my talk. Okay. So let me give some examples. The first project, remember, we are now trying to identify new biomarkers and also drug leads to target them at the same time. That's the application of my technology. So in this case, we targeted cancer cells over the normal cells directly. And so now you're familiar with the technology that I'm using. So to do this, we want to target cancer cells. And then the UT Southwestern Medical Center has the second largest cancer cell collection. And after developing this technology, they actually opened it to me. So we found two very interesting, unique cell pair. These cancer cells, lung cancer cells, and then the normal cells derived from the same patient, same lung. So it is very important to compare the same genetic background. So if it is from two different patients, that's not good. So what we did, we stained these cancer cells in red, and then the normal cells in green, mix 1-to-1, exposed to the library. Again, if you can remember, every bead has unique sequence of peptoid compounds with a lot of copies. And we are looking for only red cell bound bead. If we found something, that means the compound on this bead is binding to something found on cancer cells, which is not found in normal cells. So, and then that can be a protein, lipid, carbohydrate, or anything. And then we found one drug lead actually, sorry. And then we validated that, and it binds to the cancer cells over the normal cells. We quantify, and then now we need to go back. We don't know the target. Now we need to identify the target. So usually these are proteins in drug discovery. So the conventional method is, if you have a compound binding to a protein, you can trigger cross-linking. You can attach a cross-linking agent to the compound, and then make a covalent bond, and then pull it out, and then identify using proteomics. And we have been doing that for about a year. We waste a lot of time, and so we couldn't find anything. That means it's both good and bad. So the bad is we don't find anything, but the good thing is, if it looks like it is not a protein, it may be a lipid or carbohydrate. And we looked at the lipids, and there are 4 lipid types, and the PC and sphingomyelin are in the outer layer in the normal cells. This PS, phosphatidylserine, and PE found in the inner layer in normal cells. But in cancerous situation, this PS and PE come into the outer layer. Now, when we look at these cells, yes, we found the PSCs on the outer layer of the cancer cell, not in the normal cell. And then we checked how our compound binds. And of course, it binds to the PS very strongly, which means this PS is in the inner layer of the cell membrane in normal cells. In cancer cells, it comes to the surface. So that's the difference between these two cell groups. And our compound was able to pick that. And then our compound doesn't bind to the PC, phosphatidylcholine, which is normally found in the outer layer of the normal cells. So this is really great because our compound is very specific for cancer now. And also not only that, what known is this PS flipping happens in almost every cancer. So that can be a solution for this protein heterogeneity because the proteins are differently expressed in different cancer cells. So it can be a global biomarker. Now, I want to apologize. Now, when I show the data, I will go through very rapidly and please follow me. You don't have to look all the details. I will tell what is the important part of the every slide. Okay, when we target cell surface biomarkers, we always tend to make it multimers because if you see, like if you want to pick up something from a single finger, it is not that good. 2, 3, 4, 5, that's better. We call it avidity effect, which improves the binding. So most of the time we go for this multimerization, and actually we develop a fully on-bead synthesis protocol to develop these multimers with different linker regions as well. And I'm again not going to go into the detail. And in this compound, when we dimerize it, we found it actually kills cancer cells also, not only binding, the dimer. The monomer doesn't have any effect, and then the dimer also doesn't have any effect on the normal cells. That's great. And then there are a lot of studies that I don't have time to show, but then we jump into do the animal studies, and this is our compound very strongly accumulate in the tumor microenvironment, and the control doesn't. And also, most importantly, I'm showing the tumor reduction, tumor burden in mice. So this is the tumor's volume versus time. So the saline treatment, no drug, rapidly tumor grow. And this is our compound really working same as the docetaxel, which is one of the chemotherapeutics. And then the combination actually doesn't even let the tumors to grow. So this is great, and we are working forward on this molecule. And then on the chemistry side, once we identify these compounds, we try to identify the minimum pharmacophore, which is the most important residues that are directly important for the binding. And we find that, and then we use non-important residues to further improve the compound. For example, here we do the dimerization in the middle of the compound, and this is like we are developing a kind of a hand-like molecules. And again, they are showing further improvements. And also this is the dimer, and we develop trimers, tetramers, higher order multimers. Again, I'm not going to go into the detail. So the summary of this project is we simply compare normal cell versus cancer cell from same patient, and then unbiasedly targeted this cell surface. And we found a compound that bound to a very unique lipid, which is normally found in the inner layer of the normal cell, which comes to the outer layer of the cancer cell. And then we have done a lot of other studies. So this project is moving forward as a very uniquely targeted compounds. All right. The second example I'm going to talk to you again in the same area, which is targeting cancer stem cells over the remaining cancer cells from the same tumor. So the stem cells, you all know, they not only divide, but it can actually differentiate also. So our life started from a stem cell, and then it divided, but later it differentiate into different organs. And but we still have some those stem cells. And the problem is when these stem cells become cancerous, they become extremely stubborn because they can produce this heterogeneously populated cancer cells. And also the biggest problem is the conventional therapy mostly getting rid of only the other cancer cells, differentiated cancer cells. But these stem cells, cancer stem cells remain and they bring back the tumor. So this is the biggest problem, and the 90% of the cancer deaths are related to the tumor spread and also recurrence. And these are the type of cells that are really responsible for doing this. But unfortunately, the biology of these cancer stem cells are very less understood. And now this is the direct problem, right? When that means we don't have really biomarkers to target cancer stem cells, which is very important to target. But there are, but again, they are very heterogeneous. So the idea is, if we can get rid of these cancer stem cells, the boss is gone, so we can directly attack all the other cells and completely remove the tumor. So to do this, again, if we wait for the biologists to do this, there are a lot of complexities and there are no common agreements, no common biomarkers known for cancer stem cells. So we don't need to struggle, wait for this and struggle through, right? If we go back and think about the simple technology that I developed, what we can do is we can simply compare the cell surface of these stem cells versus the remaining cancer cells and find something specifically found on cancer stem cells. That's what exactly we did. There are, I say, toolkits to separate these stem cells. Now in this case, we separated these cancer stem cells from this H358 lung cancer cell line. And then we stain these cancer stem cells in red, the remaining cancer cells in green, and mix 1-to-1, the same story, exposed to the library, and look for only red cell bound beads. That means if we find something like this, that means the compound on that bead binds to something specifically found on cancer stem cells, which is not found in remaining cancer cells. And if it is common to both, we will see both red and green, right? So we ignore that. And then we actually found some of those after screening several 100,000 compounds in the library. And then this is the structure. And we first show that this compound is not affecting, not binding to the normal stem cells, which is extremely important. And I want to say one other fact here. Most of the drugs already, there are not a lot more, only very few, try to target these cancer stem cells. They actually attack normal stem cells as well. That's really bad. So in our compound, we found it doesn't target the other cancer cells, and also it doesn't target the normal stem cells. So, and also then we showed that when we pull down cancer stem cells, they are enriched with the some of those cancer stem cell biomarkers. And also, to cut a long story short, again, we don't know the target, right? We did unbiased selection. Now, like I said at the beginning, we have the compound with this cross-linking agent, and then let it to bind to the cell surface target, which is still unknown, and then trigger cross-linking and pull down and do proteomic studies to identify what it is. To cut a long story short, we found this Plectin protein as the target. Now, what is this Plectin? This is a structural protein found in every cell. Inside the cell, it actually connecting this actin and these intermediate filaments. Now, at this point, we have a big roadblock, big problem. Why? We did the targeting cell surface, and we found a compound that binds to an internal biomolecule. We thought we messed up, so we were wrong. But in order for this to work, the Plectin should have been on the surface. Now, when we look at the literature, we found this paper which talk about unexpected gain of function of this protein due to mislocalization in pancreatic cancer. So that means there's a literature reports showing this Plectin may be coming to the surface. And of course, that's what we found. And then this has never been reported as the cancer stem cell biomarker. That is what we found. And it is on the cell surface. And we have shown that later it is, of course, on the cancer cell surface, but not in the normal cells. And also it is found on the internal of the cell. That's fine, because we can target only the surface, right? So, and also we did a lot of studies, knock down and then look at it, and then I'm not going to waste time. And then these are colony formation assays, which shows the stem cells, when they're seeded, the other cancer cells die because they lose the cell-cell communication, but these cancer stem cells produce these strong colonies, and then actually you knock down this plectin, it cannot form colonies. And also this is actually the wound healing assay, which means we grow the cells, make a wound, and then the cells grow. That actually depict the cell migration, the metastasis of the tumor. So when you knock down plectin, it actually affects that. So that means this plectin is really an important biomarker for cancer stem cells. And then if you look at the patient data, if you have high expression on plectin, the survival is less. So that's also an interesting data. And then in the activity side, when we dimerize this compound, of course, and also it actually blocks the tumor cell migration, which is important for the metastasis. And also exactly like we did before, we did the tumor growth assay in the mice, and then we found that our compound actually can block the tumor growth, and there's no toxicity with the compound. To summarize, we find this compound targeting cell surface biomarker, again, complete unbiased selection. And we found this plectin as a biomarker for cancer stem cells. And we have a compound to target it. And we're already showing that it has activity against these most difficult cancer cells that we cannot control easily. And also the cool thing about our technology is the normal proteomics and other technologies cannot find these proteins moving from internal of the cell to the outside. We were able to pick that. Okay, actually, when we published this in the Nature publications, the scientific reports, in 2019, actually, this became one of the top 100 downloaded articles, and we recently published the activity also. Let me talk about the third project. Again, the same unbiased application. Now, this time, we are going to transform cells from normal to tumor and try to identify early biomarkers for early detection. So this shows the tumor growth, and then it's exponential after that angiogenesis, after new blood vessel formation. The current threshold of the detection is after the angiogenesis, which is the tumors very strongly started spreading. So can we get this down? To do that, okay, so the tumors doesn't grow overnight. It takes months, years. So there are a lot of things happen at the beginning to the progression, and then the angiogenesis happen and go to the latter part. So the current detections are here. Now, if we can detect the tumors here, we can address a lot of and save a lot of lives. So to do that, we need to find these earliest biomarkers. Of course, there are a lot of effort to do this, but the conventional approach again, all right, we have a list of biomarkers here, and then what researchers do is whether they are found in early cancers. But again, that's bias. So what is the unbiased way? So we were very lucky to get again this very interesting cell lines, which is starting from a normal cell, introduce these most important tumor forming mutations one at a time, P53, KRAS, and then the c-MYC, and then convert these cells into full tumors starting from normal. Now what happened is we hypothesized that from the normal cell, when these mutations happening inside the cell, new biomarkers start appearing because the tumor wants to do many things. And these new biomarkers are the earliest biomarkers. Now, can we pick them? That's the great question. Yes, we have this technology, this On-Bead Two-Color technology, which can compare two cell groups and very accurately find out the differences between two cell surfaces with this red and green color application. And that's what we did. We can compare normal to this one and normal to this one, normal to this one. And then find, we did the first, normal to this fully transformed cells. And this is the assay. Again, I'm not going to go to detail. We found a compound. We showed that it binds only to the tumor cells, not to the normal. And to cut a long story short, we pulled out, we've identified this compound, actually this target as vimentin. Anyway, that was known before. So what happened is this vimentin, again, mostly found in the internally, come to the cell surface. The most studied part here is in the metastasis, tumor spread, but we found, and also it has been reported to be found in the early stages as well. We found it is in the very, very earliest stage in the cell surface. So, and we have a compound to target that too, because we, in our technology, we not only identify a compound to target the biomarker, but we identify both biomarker and a compound to target at the same time. So that means it can be used for attach and deliver contrast agents, imaging agents. You can use for diagnostic and also for therapeutic applications as well. So in the diagnostic side, we are very excited to see this. We already use this for the immunohistochemistry. Now, when we get the biopsies from patients, this bottom panel is the normal way of doing, which is the using antibodies to do this in these reagent kits. But we can replace those very expensive and very difficult to handle reagents with very simple, straightforward chemical compounds. And this is how our compound works. The first 4 panels are the cancer. The last one is normal. It's a very clear demarcation between cancer and normal cells. We are taking this forward with this, again, this patented compound. And also we are trying to develop very simple diagnostic assays, which is still, we need to do a lot of optimizations. The whole idea is to, can we develop a simple diagnostic test that can be used in the doctor's visit in the normal clinic. But we are not there yet. There are a lot of things to optimize. Okay, like I said, we can identify the minimum pharmacophore, and then we can use those important residues to fine-tune binding. Non-important residues can be converted to new binding, and the new moieties can be added like this shown here. These are the actual structures of these compound improvements, and then I'm not going to go into the detail. And then on the active side, which is the therapeutic side, the dimer actually shows much stronger activity. In this case, actually, we pick cell lines, cancer cell lines, that are with high vimentin, moderate vimentin, low vimentin, and no vimentin. And you can see the cytotoxicity assays. The high vimentin has the highest effect. IC50 low means both highest effective, and then the moderate less, and then the higher low vimentin means no activity. So that's great. It is targeting vimentin and bring the activity. And again, the compound destroy these colony formations and also migration. All great. Okay, so please give me a few minutes to talk about this very last project, which is very interesting, and it's targeting COVID. So when the pandemic happens, we were also thinking to do something. And with having this very interesting, cool project, the technology, we were thinking about targeting this virus targeting biomarker. So we all know the virus comes in and goes through the nose, and it actually targets this human angiotensin converting enzyme 2 receptor, ACE2 receptor on these epithelial cells. So this virus has this spike protein S, and that spike protein bind to this human ACE2 and then get into the cells. Now we thought about, how about we develop a molecule that can block this interaction, and then it simply blocks the virus entry. And it can be the molecular version of the mask, simply putting the mask as a molecule in the doorknob, right? When the virus try to get through the door, if you block the doorknob, it cannot get in. And then it can be used as a nasal spray or eye drop. And this is very simple, straightforward, right? I mean, it's not a big idea, but no drug, this kind of a drug has been developed so far. And not only COVID-19, SARS-CoV-2, the other SARS viruses also target the same receptor. So that means this mechanism has been known for about 2 decades, but no drug is there. The biggest reason is this. This ACE2 receptor is very important for humans. So basically, this is what we're going to do. This is the crystal structure of the ACE2, and then the virus has protein binding. This is the interface. We are trying to put a face mask there as a molecule. But we need to pay attention to this area. So what is this ACE2 receptor? This ACE2 receptor is the one that actually regulates our blood pressure. So if we do anything to this receptor, it actually messes up with our blood pressure. Now, this is the structure of the ACE2, and this is the virus S protein binding, and this is the active site of this ACE2 receptor. And I said this has been known for more than 2 decades, and there are many compounds have been reported to bind to this, but none of them become virus blocking drug. The reason is all these compounds actually affect this active site, and therefore it becomes toxic for us. So, but when I look at this assay, again, this is another take-home point. Think simply. All the compounds that touches this receptor seems to be toxic, but virus does that, right? Virus cunningly come in and touch somewhere on the surface of this receptor without affecting this area and cunningly get into the body. How does that happen? So my hypothesis here was how we mimic that, how we, in the evolution, the first day when the virus came in and saw this ACE2 receptor and it touches some place. And that place was the perfect place without affecting to the cells to get in. And that's what we did. So if you look at our cell screen, so we had ACE2 positive cells and ACE2 negative cells, red stain, green stain. Now you know we are targeting this cell line with the ACE2 shown here. Now if you pay attention to this area, when these compounds are display on the beads, it's just like virus, right? Coming and to see this ACE2 receptor. So we exactly mimic that situation and identify 2 compounds and also further optimized it. And we show that it is very strongly binding to the ACE2. And also we showed that it actually blocks the virus's protein binding in biochemical level. And also, most importantly, we show that the compound actually blocks the virus entry into the cells as well. That's really great. And even with some mutated versions. So the biggest question is the toxicity. So we went back and did a lot of studies to show that this is the cytotoxicity assay. We found 2 compounds out of 50,000. Both of them have no effect on the cells, no toxicity. And then no effect on the ACE2 protein. It doesn't do anything to the protein. And also, it doesn't, most importantly, we did the enzymatic assay in many cell lines and it doesn't affect the ACE2 enzyme activity. That's really great. And then we did some molecular docking studies. What we found was exactly as I hypothesized, this compound really bound to the exact place that the virus has protein binding. So it actually blocking the interface simply as exactly like we put this molecular mask. So we have this, basically the molecular mask that can block this ACE2 virus interaction. And we are actually working with developing a company and taking this forward. So the idea is to develop nasal spray or eye drops like applications and then to use it in the morning and to go to work. So, but again, I want to again re-emphasize, I have not developed a drug yet. So these are still in the research and development phases. All right, so I want to summarize all these projects, and I mentioned my main aim is to kind of look at this conventional drug discovery process and try to shorten it or make it rapid. So the processes are initial compound identification, validation, optimization, and then go to the preclinical animal levels and then the clinical studies. So these are the several different projects, actually 8 of them have been patented. All these compounds were brand new, completely new compounds, and the green means what we have already done. So we are working on different levels of these projects. And then finally, I want to thank all the people who have done this work, and then the collaborators throughout the US and also worldwide. And then most importantly, funding, the NIH, Cancer Prevention and Research Institute of Texas. And these are the two major funders for me in multi-million-dollar grants. And then these are the Owens Foundation, Rheumatology Research Foundation, and also our own University Drug Discovery Institute. All right, so I will stop here. And if you have any questions, anything I can answer. Thank you so much for your patience.
Thank you, Professor Gomika Udugamasooriya. So I think you have plenty of work starting from cancer research to COVID-19, quite interesting. So I think our listeners will have more questions. So now the floor is open for questions and clarifications. So let me begin with the AC2 enzyme, the related to COVID-19 research. So in your docking studies, have you ever noticed that the binding affects the chemistry in the active site? Does it has an effect or not?
No, that's what I said. We did very careful studies because all of the previous compound failed because of that toxicity. So we first did the direct enzyme assay, ACE2 enzyme assay. There are kits to take that, and with higher concentration, even with higher concentrations of our compound, which bound to the ACE2, still the ACE2 activity was fine. And also we did the studies in the cells for the activity, no effect, and the compound binding doesn't affect the protein structure, or it doesn't destroy, and also it doesn't affect the cell viability as well.
Thank you.
Thank you very much, Professor Gomika. I'm Dr. Jagat. I have one question about your peptoid, because I'm also worked on some peptoids.
Okay.
I want to know whether your peptoid is going into the mammalian cells or it just stay outside.
Oh, actually, that's a great question. So the inherently peptoids are cell permeable. You know, peptides are not that cell permeable because they have the amide bonds. The hydrogen bonding happens, so therefore the cell permeability is reduced. But if you can remember, in peptoids, the R group is on amide nitrogen, therefore it is more inherently cell permeable. So I didn't have time to explain a lot, but we are targeting cell surface, so we prefer it to stay there. If you can remember, all the projects, we immediately went for multimerization. So this multimerization actually do multiple things. One is, again, as I mentioned, improve the binding because it can bind to multiple copies of the targeted receptor on the cell surface. But at the same time, it actually reduces the cell permeability. You know, it targets whatever the target found on the cell surface and then do the function there. I don't know, is that what you meant?
Yes, yes, I got the correct answer from you. And have you tried with prokaryotic cell membrane compared with the eukaryotic cell membrane?
That's a great question. Yes, so we really want to get into that. Actually, so we have not done it yet, but especially with this lipid-targeted compound, because there are a lot of interesting things can be extracted, and yes, we really wanted to do that. And again, with so many different projects, and what happened with my research program is like, usually when you work on, you have your most interested target and you have most interested compound, and you work on that, right? But in my case, not only me, there are a lot of requests for the collaborations, and everyone brings their target to find a compound. And my own, I have already 8, 9 different targets in the systems, right? So it's good and bad. So I definitely want to do that. That's yes.
Okay, thank you very much, Professor Gomika.
You're welcome. Any other questions?
Do you have any more questions?
I would like to hear from undergraduates. Remember, come forward. And I told you my story at the very beginning with my great master, Professor Dilip De Silva, right? So when I asked that question from him at the very first lecture, and that triggers a lot of things, and I was able to very well benefited from that interaction, right? So please come forward and ask questions. So don't be shy, and you have to do this if you want to develop a strong career in the future. You can sit down in the back row and still get the 4.0 GPA, but that doesn't make you a really effective person in the research and also in the career building.
Dr. Telmat, may I ask a question?
Yeah, go ahead.
Yes, go ahead, Dr. Lyra.
First of all, Dr. Gomika, thank you indeed for being here. It was a very splendid experience listening to your presentation. It was inspirational. That's exactly what we wanted to hear, sir. Unfortunately, I'm not an undergraduate, but I thought of asking this question because the whole objective of us having you here today is to motivate our younger generation, because we believe that we have a lot of potential, but we need this driving force, the impetus for the younger generation to find their actual inner potential. So what would be the answer if I ask you the question, how did your background in Sri Lanka and the Sri Lankan education system take you to where you are today?
Okay. If I begin very honestly, we have a big problem in the system. I think that's a big debate. I don't want to get involved into that. But anyway, so, okay, again, I can begin from there. So everybody is talking about that, but you blame outside for that, right? That means you are waiting others to correct the system, right? But I challenged the system and I came out of that and I'm here now, right? So even like if you go back to I didn't have a chance to explain all this, and I didn't get selected for medicine, right? In the biggest topmost thing in Sri Lanka, the biggest dream, right? But I'm here now. How come? It's because the same exact thing that I mentioned, the interaction with in the first lecture. I told you, I'm telling you this multiple times. That's what we are not doing. So when I came forward, I didn't sit down on the back row of the class, and I was in the front row. I was interacting with faculty and answering questions, coming forward, and I found my way. I think I can summarize by saying that you need to come forward, and again, like I said, with that, the way I impress Professor Dilip, actually, sir has given me a lot in my career. So if I didn't do that, he wouldn't have recognized me, right? So the same way, you need to come forward. There are a lot of great faculty, teachers around, and then interact with them. And then again, don't be shy. Try everything. And you see a lot of obstacles, a lot of problems. But if you are coming forward, you can find the way. And also in US, I mentioned my discussion with my postdoc advisor. And remember, he directed me to become a really independent person. And even I was just trying to do something slowly, or like kind of elevating something from this level to a smaller level. He said, no, if you want to go to that level, you need to try something brand new. And I took it very, very seriously. And that's how this whole technology become reality, right? I mean, and now it has been used in very widely in everywhere. And so again, my short summary again is, don't sit down and blame the outside and nothing is happening. Oh, okay, you cannot change the world easily, but if you try to adjust and then go around the obstacles and find your way. I don't know, I told many things, but there's I hope I gave some thoughts for that question.
Yes, indeed. Thank you very much, Professor Gomika. That's actually kind of what we wanted to hear. And without further ado, Professor Gomika, we have a very special guest here with us today. I believe you might be able to see that person on screen right now.
Oh, oh my God. Professor Dilip is here. Okay.
Hi, Gomika.
How are you, sir?
Really nice to see you. I enjoyed your talk, and I think you are a real good model for all our students here. I think in our universities, we have great talent, really great talent. And we see them. Unfortunately, a lot of them leave Sri Lanka, but that's okay. They are serving the humankind somewhere. Unfortunately, I'm very sad to say, probably if they stayed in Sri Lanka, they can't do that. So like you said, we have to change our system. First, as you say, people has to start talking. Okay, you have to express yourselves and question, and also our mentors also have to change their attitude and encourage more people. Anyway, it's great to see you, Gomika, and keep up the good work. Okay.
Thank you so much, sir. I mean, you actually changed my life.
I don't think I did anything much. Okay, thanks anyway.
Actually, yeah, I'm really glad that you are here, and then actually it confirms what I was telling from the very beginning. And I shared with everyone about how we interacted first, and again, it's both sides, like you mentioned. Now,
Yeah.
as a person who is sitting and directing students, and so my approach also in the working with the both PhD students and also undergrads and even the postdocs. So my approach is at the very beginning, I guide them, but very, very rapidly, I want them to take over. So one of the biggest things I do for them is from day two. Day one, I'm actually guiding, but from day two, I want them to come forward and do their work and plan things, ideas, and take them forward. And that's what you did for me, and that's what my postdoc advisor, PhD advisor did to me. So that's how actually exactly like you mentioned, the faculty, the advisors also have a big role to play. And again, we definitely need to change the system, because there are a lot of talents being misplaced or dropped on this system. So even the faculty individually try to do this exactly like you did. And then if the students also come forward, we can make use of our potential.
Yeah. No, I fully agree with you. There's great talent here. Great talent. So,
Yes.
and University of Colombo, I'm sure all the other universities have produced really some great students who have gone abroad and doing. Some of them, of course, come back. That's fine. And some people tend to think, well, going abroad, well, they should be back here. No, I think anywhere in the world, it's okay. As long as you do work, that's what counts.
Yes, I really want to come back, and you know,
I'm sure you would like to come back here and serve your motherland. Yeah, I know that very well. Yeah, okay, Gomika, nice talking to you.
Thank you very much, sir.
Thank you very much, Professor Gomika, as well as thank you very much, Professor Dilip. Over to you, Dr. Thelma.
Yes, thank you, Dr. Lahiru, and thank you for all the participants too. And to end up the question and answer session, I think I should take up something from Professor Gomika's webinar today. So according to you, sir, you suggested us to start simple, think simple and start. So I think that's a great start for many of us, including the students as well as academics, so that starting something simple, we thought like it's not going to be the scientific way, but really it paves your pathway in research. So thank you very much for sharing all your knowledge today. But to officially introduce the vote of thanks, I would like to call Dr. Dinusha. Thank you, sir.
Can I give a minute to comment on that? Yes, think simple is the most important part, but you need to have a big goal, the big scientific question. But to answer that, you need to think very simple. And actually, this is a simple red and green two-color assay, which is a kindergarten thing. But until I developed this format, over 2 decades, this format of high throughput screening didn't apply. And it's a very simple thing to do. There were some technological things to improve, but I can share one other experience also. When I try to publish this work, and I use the word, it can bind to something on the cell surface, on the cancer cell, which is not found in the normal cell. And the reviewers and the editors repeatedly told me to remove that word. It is not scientific, saying, when I say something on the cancer cell surface. And actually, I fought with them. I said, yes, science means not catching the biggest thing. We can use anything, and then actually that's how you need to approach. Thank you.
Yes, thank you very much for sharing your experience. So Dr. Dinusha, it's over to you now.
Okay, thank you, Dr. Thelma. On behalf of the Section E2 of Sri Lanka Association for the Advancement of Science, it's my privilege to propose a vote of thanks to all who contributed to the success of this webinar on Sri Lankan Chemists in Focus. Firstly, I would like to express my sincere gratitude to our resource person, Professor Gomika Udugamasooriya of the University of Houston, USA, for sparing his invaluable time to grace the occasion despite his busy schedule and enlightening us with his knowledge. I'm very sure the audience were very much interested on the information delivered on novel chemical biology technologies to expedite the drug lead discovery process. Especially the story you shared was very interesting. Thank you, Professor Udugamasooriya. Secondly, I would like to express my earnest appreciation to the General President and the Council of SLAS for allowing us to conduct this webinar. Thirdly, I would like to extend my thanks to the members of SLAS E2 committee, especially the organizing committee, for the continuous support they have given to organize such a valuable and informative session. At last, but not the least, I would like to thank everyone who participated for this webinar, and we hope to see you in the near future with another webinar under the webinar series of Sri Lankan Chemists in Focus. Thank you. stay safe, and have a pleasant evening. Over to you, Lahiru.
Thank you very much, Dr. Dinusha. It is indeed a very pleasant evening, and it's a pleasure to be here. And it was a very enlightening to listen to your presentation, Professor Gomika. So once again, on behalf of Section E2, I would like to take this opportunity to thank you for being here, Professor Gomika, and moreover, for accepting our invitation to be here, sparing your valuable time. And I'm sure, just like you mentioned, sir, there's a lot of potential in our students, in our next generation, as well as our current generation. We have a lot of potential. It's only the fact that they don't see the inner potential. So we need to make sure that as academics, we try our best to take out the best of our students, just like what you experienced. So this effort was all about it. So therefore, we were expecting a higher number of undergraduate students to show up. Unfortunately, as you know, probably, sir, we are experiencing a lot of difficulties right now in Sri Lanka, especially with the power outage. So therefore, most parts of the country are right now experiencing a power outage. So therefore, with that current situation, unfortunately, we did not receive enough undergraduates. We did not have enough undergraduates present in this session. However, I'm sure whoever was present will take the maximum benefit out of what you had to say, sir. And of course, we have recorded your presentation. With your consent, we will definitely upload it to this last web page as well as this last YouTube page. So therefore, that will definitely benefit a much more wider audience as time would permit. Before we wind up the session, I would like to make one more request from the audience. I'm sure everyone had a lot to learn from this delightful presentation. So therefore, I would like to request you to turn on your cameras, because I mean, our interactions have been limited to a virtual platform, unfortunately. But to make it at least a memorable occasion, let's turn on our cameras and try to capture some memories for us to cherish during the year. So can I request all of you, anyone who is comfortable, to turn on your cameras?
Definitely, and I really would like to see that, my fellow scientists in Sri Lanka. So, and also, please, this is again for the undergrads, please don't stay back, come forward.
Thank you, sir. Let me capture some screenshots. Yeah, we are waiting for a few more people to turn on their cameras, hopefully. Okay, let me check. Okay, looks fine to me. Thank you very much for everyone for turning on your cameras. I mean, these kind of little things will definitely make a lot of difference. And just like you rightly said, Professor Gomika, I think one of the key reasons that we as a nation has led back is because our tendency to speak up in public. So I think that is definitely something that we have to inculcate in our future generations. We have, from a very tender age, we have to make sure that they are comfortable talking in front of an audience and speaking up for their rights. So that's definitely something that we have to do as academics as well. So we are doing a lot of activities in that regard, especially when it comes to universities. I'm sure now the system has changed a little bit from where we were a few years back. So we are trying to get people to participate more in the classroom. So the typical classroom-led lectures are no more, hopefully. So therefore, we are trying to get them into a discussion rather. So hopefully with that trend, we might be able to make some difference in future. So that's just being hopeful for the future, sir. But like you said, I think there's a lot to learn. But the bottom line is we all believe that we as Sri Lankans have a lot of potential. So therefore, seeing people like you is definitely inspirational to our younger generation. So Professor Dilip and Professor Gomika, both of you have been an inspiration to all of us. So therefore, continue the good work that you have done to many more generations, to the mankind, as Professor Dilip Disila said, wherever you are serving the mankind. That's the most important thing. So therefore, we wish you all the very best to continue the good work with everything that you do. Thank you very much once again. And before we wind up, a couple of reminders. We as Section E2 are organizing many activities. Let me quickly share the screen. So this is one of the activities that we have organized during the current year, and To brief you about the upcoming activities, during the month of April, we are planning to have 2 more webinars. The first webinar is by Professor Ravi Silva from the University of Surrey. He's going to be conducting a webinar on energy transitions to greener and cleaner technologies. Definitely something that's contemporary of importance. The second webinar is going to be conducted by Professor Nalin D. Silva. This is partly one of the webinars in the series that's titled as the Pathway to a Successful Career in Chemistry. So that once again is specifically oriented for our undergraduates who are studying chemistry. So these activities will be in line once again during the month of April. And if anyone in the audience would wish to stay tuned with the activities that we are planning for the future, please join our WhatsApp group. As you see, there's a code displayed on screen right now. If you scan this QR code using your WhatsApp camera, you'll be able to join our WhatsApp group, which will enable you to stay tuned with our activities lined up for the year. So that's all that we have for you. Thank you very much once again, Professor Gomika. Wish you all the very best with all your future endeavors. On that note, let me wind up the session. A very pleasant evening to all of you. Thank you very much.
Thank you very much again for the invitation, and all the best for you all. And I know it's a very hard time going on Sri Lanka, but again, as an individual, try to make a difference.
Thank you very much, Professor Gomika. Is there anything to
Thank
add?
you. No, actually, again, I mean, we want to reiterate that there are a lot of changes need to happen definitely in the education sector, and we have been talking about that for a long, long time. But again, as an individual, until those are happening, those will happen somewhere in the future. But you can do what you can do. That is the message, rather than, you know, if I say in Sinhala, අර කියමනක් තියෙනවා අර අන්ධකාරයට ශාප කරලා ඇස් වහගන්න නැතුව එක ඉටිපන්දමක් හරි දල්වන එක වටිනවා කියලා ඉතින් so please remember that.
Thank you, sir. That's
Thank you all and all the best.
Thank you, sir. Thank you very much. We look forward to your cooperation in our future activities as well, which I'm sure you will be happy to be a part of. So thank you very much for
Definitely. Definitely. Actually, I forgot to mention that please let me know if you have anything, and I can be supportive and helpful, because Dilip sir know what I have been doing, and there are a lot of things I have been trying to do and doing also, but definitely for these kind of things, the workshops or whatever, whenever you need, let me know, because I never forget the Sri Lankans actually funded me for my education and the free education. So I always want to pay that back. And so please let me know whenever you have anything that I can be useful. Thank you.
Definitely. Thank you very much, sir. Okay. So on that note, I believe we have taken a lot of your time, sir. So we'll end up the session. Thank you very much once again for everyone for being here. We hope to see you once again in the activities that we have planned in future. So have a very pleasant evening and all the best to all of you. Thank you very much.
All right, best wishes.
Okay, thank you, Professor Gomika. Thank you very much. It was really nice to from the invitation, you kept on replying. So it was really good. Thank you very much.
Thank you, Dinesha. Thank you. And all the best for you all.
It was very nice to see Professor Dilip after so many years.
He's muted.
Yeah, maybe you can stop the recording.
Yeah. Okay.
Was it who was just talking actually?
Amila.
Amila.
Oh, okay.
Yes, it's me.
Okay.